Hello friends, I welcome all of you to Manan's e-learning program. Before I begin my lecture series, the lecture number 2, I would like you to attempt this MCQ-based questions. So you will see some of these questions. You just mark your answers. So I welcome this. In today's lecture, I am going to talk about the terminologies in genetic lecture. I hope you get benefited from the questions. It is just as a practice. For those who do not know me, my name is Nan and I am a PhD in molecular biology. In today's lecture, I am going to introduce to you certain terminologies which is often used in genetics. In my previous lecture, that is lecture number 1, I've already described about what is introduced the subject of the genetics, who coined the terminology, and as well as Mendelian genetics concept. In today's lecture, we will be taking some definitions. I will try to describe you monohybrid cross. I will also describe you backcross, test cross, and Mendel's principles of independent segregation. And in some places, you also find out the principle of dominance. So please stay tuned. So this is a definition which is being used to define the genetics concept. Now, genetics is a study of the genes or the genetic variations and hereditary of an organism. It is often also called as a structure and function of the genes. If you want to know more about this, so you can follow this link, which I will also will be sharing with you, and then you get benefited from this. Now, the first definition which I would like to introduce to you is the alleles, and in a very simple form, it has been written that it is a different forms of a gene. Now, we know that the genes which are present are on the chromosomes are a stretch of the DNA. Now, we also know that as we received or any diploid organisms has received one chromosome from the father side or the paternal side and one from the maternal side. So let's say this is one chromosome. I'm trying to draw here. So this is one chromosome and this is another chromosome. These chromosomes, one is received from the mother side and the other one is from the father side. These chromosomes are also called as homologous chromosomes. Now the gene which is been present over here, the other gene is present in the exactly at the same location. Let's say we define this with a letter capital A, then there is will be another capital A which is in exact position. So technically, if we have to define, there is two genes which is been present because of the two chromosomes in the diploid organism. There is a possibility that this gene A and there is a slight difference in a gene which is denoted by small letter small a. These are called as the alternative forms of a gene. Let's take an example. Let's say there is a plant which is tall and we define its genotype with the letters capital T and capital T. There is a plant, another plant which is short in length and we denote its genotypes between a small t and t. So we also trying to know what is a genotype and what is also a phenotype. So the phenotype is basically the physical appearance of a plant. Okay, let me show you. in a moment what I meant here. So if you see, this is the Mendelian Mendel, which he used for his studies, the height of a plant. So this plant is tall. This plant is a short plant, a dwarf plant. Okay. Now coming back to my previous slide here. So we denote this is by genotype. So the physical appearance of it is called as phenotype, and with the symbol letters in genetics we use this as a genotype. Now uh, if these two plants is being crossed, let's say plant 1 and plant 2 is being crossed, the next generation which it's come is called as the first filial generation. Now this progeny or this offspring will receive one chromosome from this and one chromosome from this. We can define them this is a female plant or a male plant over there. So this offspring will receive a capital T and a small t over here. Now this particular individual, now if we have to define the chromosome, if we have to draw this chromosome again, and I'm trying to explain the concept of the alleles, and this is a position and this denotes is by capital T, and the exact position you have an alternate form which is called as a small t. It also leads to another definition which we call the capital T and T as an homozygous plants, because they have the same types of the alleles, or a small t plant is also called as a homozygous. But this individual which is having two alternative forms of the genes or two different alleles if you want to say, then these are called as heterozygous. Okay. Now the another terminology which is often used is the character. Now these are basically the characteristics of an individual that is been transmitted from one generation to another generation, and it is often used as a synonyms for the trait. Now coming back, taking you forward, that these 7 characters which is being studied by Mendel. Okay, now let's say we are taking examples of the plant height. I can change the color code so that it is visible. So plant height, and we define that this is the genotype of these two plants, and phenotypically it looks like this, tall and small or short. The progeny of these two cross of a plant, we also saw that this is a heterozygous individuals consist of the two different forms of the alleles over here, right? Now all these characters, the phenotypes which you are mentioning, they are also called as the characters. Okay, and the two alternative forms, that is a tall and short, is often used by the terminology which is we call as a trait. Okay, so coming back here, so this is how we defines the alleles, and this is how we defines the characters. I've also introduced to you the concepts of the genotype and as well as the concepts of the phenotype, and I also defined to you the heterozygous and the homozygous individual. I've also introduced a concept which is called as the F1 generation and the F2 generation, which is the first filial generation and the second filial generation. That is the progeny of mating of an individual of the P generation, that is the parental generation, and examples which we took, that is P1 and P2, is called an F1 generation. Okay, like if a man who is been married to a woman, okay, and all the progenies or which is there, these will be called as the F1 generation. Like I can be considered as an F1 generation for my parents. Okay, that's the personal example I'm taking, but to make you understand. Okay, similarly, the offsprings which I am having, those will be called as a second generations, and this is a second filial generation. You often heard this also in that someone who is grandparents, those are the parental populations, and then there is the first generation is their children's, and then they have another children's or the grandchildren's, those will be called as F2 generation. It is as simple as that. Okay. Now we also come across a terminology which is often used, which is called as a gamete. Now, gamete is a mature reproductive cell that is specialized for a sexual fusion. Now, if I took this as an example here, and let me first rub it so that you can be able to understand that what I am trying to say, how we basically talk about the gametes. Now, the gametes, like for example, there is a plant P1 and it has been crossed to plant P2, having a genotype, let's say capital T and small tt. Now, to define, because this is a diploid individual, or you can say they have the two chromosomes over here, okay, like this. This is another chromosome for this particular individual, and they have these genes which is been present and denoted by this letter capital T and small t and t letters. Now, during the process of the gametogenesis, okay, or which is basically depending upon the sexes will be called as spermatogenesis or the oogenesis processes, the total number of the chromosomes becomes half. Okay, so let's say as a human being, if we take an example, okay, now as a human being, if they have a 46 chromosomes, and let's say this is a female, and there is a, let's say, another individual which is a male. Now, during the, she will be producing the egg cells. Okay? The egg cells which it will be bearing, whatever the number of the egg cells she will be bearing, this will be reduced to half. So each of these egg cells will consist of 23 chromosomes, not 23 pairs. Okay, not 23 pairs of chromosomes, 23 chromosomes. Similarly, a man who will be producing the sperms, these sperms which will be, let's say we denote these sperms like this, these sperms will also consist of 23 chromosomes and not 23 pairs of the chromosome. That's how it is been made into half. And the process by which it is being made, this process is called as meiosis. Okay? The meiosis. So how basically it works? So we denote this by a symbol that any diploid organisms by 2n. In some places you will also find it is defined as 2c. So the 2n number is 46. And what is the n number then? The n number is the 23. So which is the haploid sets of the chromosome. This is called as the haploid sets of the chromosomes. And as a diploid, so we multiply 23 by 2, make it 46. But during the process of the meiosis, or in the gametogenesis in specifically, say this first things which is doing, it occurs in the cell cycle, and in the cell cycle process, if we have to define, maybe a separate lecture I will use to describe meiosis, but to explain the principle of segregation, I have to give little bit background, and that's what I am trying to give it to you. So there is a G1, there is a S, there is a G2, and the actual process of mitosis or meiosis which is taking place. So in this process, that is the S phase, there is a duplication of the genome take place. Means if there is a 46 chromosome, so initially they will becomes 92. So this 2n number, because if you remember this, the n number here is the 23. So now it will become as an 4n. And after this 4n, it will be in the first division, which is meiosis, this which is called also reductive division, will be equally divided as 2n numbers, and from these 2n numbers they will be made as n, that is the haploid sets, and similarly this is also made as an haploid set, that is the n numbers. This is how the gametogenesis process work, and these sperms or the eggs which is harboring the half number of the chromosomes, these are called as the gametes. Now during the process of fertilization, so this is an egg cell, let's say, and this is your sperm. it comes and fuse. So it is having n number, this is having an n number, and then it's basically fused to make a 2n number or a zygote. This zygote further divides from one cell to two cell, four cell stage, and then to up to 100 cell stage to make it is a blastocyst. From this blastocyst, then it becomes the morula. Then from this morula, it's basically the process of embryogenesis starts. Then it gets implanted into the uterus of the mother. Then placenta starts coming, and that's how the individual basically works. What is worth to mention here, or one should take home lesson which I would say here, is that each individual for the eukaryotic systems, they receive 50% of their chromosomes from their mother and 50% of their chromosomes from their father side, or the maternal and as well as the paternal sides. I hope that clears my point. The next terminology which is we are already defined, that is gene. So I've already defined that these gene, but for the Mendelian genetics, it is also called as Mendelian factors. He named those as a factors because at the time of Mendel, he was not aware of the concept of gene, DNA. He is not known to them. So he told them that these factors which determines the characteristics of the organism, and then he used those symbols to represent them. Like for tall height, it is capital T, or say for the round shape of a seed, then he uses a symbol like capital R and small r and so on. So we will be talking about that in a moment. And the genotype, as I mentioned, this is the genetic constitution of an organism, and the phenotype is the physical appearance of the organism, which basically governed by the, so we can say the phenotype of an individual is governed, let's say it is phenotype, is by the genotype plus the contribution from the environment. Okay, now this is very important here, because whatever we are, it is not only because of the genes but also due to the environment. If a gene it does not express, then there will be a problem with that. Okay, let me give you a real life examples to make you understand. The peoples who lives in cold areas, okay, they often the vitamin D's has been supplemented in the milk over there, and the reason for that, because they've been not exposed to the sunlight. And it has been observed, the studies has been observed, that due to the vitamin D from a very early age and the weather conditions, their height is comparatively much higher as compared to the person who is in a warm region. However, there is a more details is been required, because again the height of in humans is a polygenic inheritance. It is not simple as that. But what I'm trying to make you understand, that there is a contribution from the environments does takes place, and it is mostly associated with the gene expression and the DNA methylation and so on. Okay, so maybe in some other lecture we will discuss. The essence of today's lecture is to make you aware of the genetics terminology. Okay, I hope it is so far so good. Let's move ahead here. So these are the characteristics or the characters which were studied by Mendel. We defined this in our previous lecture number 1 as well. Now you see here is, that is the two things has been mentioned. Okay, like say for example a seed color has been mentioned here, or a seed shape, a yellow versus green, or a smooth versus wrinkle, or a height of a plant which we just took, tall versus a short height of a plant. Now these are two contrasting forms of the phenotype. Now to understand this, we will use a cross, and this type of cross is called as a monohybrid cross. When we say monohybrid cross, we are interested in knowing only one of the characters at a time. Okay, what is a dihybrid cross? I will take this is in my lecture number 3. But right now, just focus upon what is a monohybrid cross. Let's say there is a one plant which pod color is green and another plant which is having a pod color of yellow. The genotype of this particular plant we can define as capital G and capital G like this, and say for the yellow, for the simplicity sakes, we refer to as a small g and small g, just to differentiate the genotypes of this individual. Phenotypically, they are green and yellow. Now, what Mendel actually did in one of his cross, that he took this as one of the plant and then he crossed this plant, that is a green pod color of a plant with a yellow pod color of the plant. So we expect that is the next offsprings, that is the F1 offsprings. Okay, that is how a genetist wants to know that how it is, and Mendel was trying to understand. So to his surprise, he found that all the F1 plants were green pod color. Okay, green pod color of the plants he observed. Now he used certain letters to make it understand, and this is basically what we are trying and know these things, that is called as the process of the gametogenesis, which I explained to you in a moment ago. Now in genetics, if we want to say that this particular offspring has received 50% of the chromosome, so we are actually interested in knowing only one character or one trait over here, that is the green, so that's G will be received. Similarly, this particular will also parent will also contribute, and a small g, so this individual will be heterozygous. We call them as a heterozygous, and this individual we called as an homozygous individual. Okay. Now if we say that the genotypes are capital G and small g over here, why it is green? Mendel did this experiments for his another study, like he take the height of a plant, and for this height of a plant he take a tall plant and a short or a dwarf plant. Now this parental population P1 will be capital T the genotype, and it is crossed with another plant which is P2, which is denoted by genotype small tt. Now in the F1 generation, he observed all the plants are tall in height. That's what he observed. Now again going into our definition, that 50% of this chromosomes which will be received from the male or female, doesn't matter, plant P1 and the small P2 is a heterozygous individual. So you observe all the plants is the tall over here, and so forth and others. So let me first rub it, and so that I can explain from this picture as well. So what Mendel did, that he took a green, so the green is coming. When he took the inflated, and with the pinch he found that inflated is coming in the F1 generation. When he took the yellow and the green seed color, he found that yellow is coming over here. When he take the seed shape, he found that smooth is coming. When he take the flower color, the purple is coming, the tall is coming. Based on this, he found, or he proposed, that one of this character is dominant. Okay, that is dominant. I will share one video link with you. This is from TED Talk, and this link they have also very elegantly explained this phenotype of monohybrid as well as a dihybrid. So you can also watch that in the link over there. But right now we are trying to understand what is a monohybrid cross. A monohybrid cross is a cross where you are taking the characters one at a time. Say height of a plant, the shape of a seed, the color of the seed, the flower colors into consideration. Now in F1 generation, we find all of these plants is to a one particular phenotypes, means they are all appearing to be green or they are all appearing to be tall over there. And he said that he tried to explain them that it is basically one is dominant the other characters. The other one which is one is a dominant, the other form is called as recessive. We will understand this more. We will discuss this our my subsequent lectures as well. Now, I would like to mention here the back cross and a test cross. What does that means over there? Before trying to explain to you the principle of segregation. Now, it is very easy to understand. Like we take our same example, that is a pea plant, that is plant 1 is crossed with plant 2. Let's say this is, take another example. This is a round shape of the shape we are taking, and this is a small r plant which is having a shape wrinkle over there. So in the F1 generation, we find that the genotype of this plant will be capital R small r, and they all will appear as a round. I would like to also mention here the way the genetics often use a Punnett square, okay, to solve this thing, and it's very easy to follow. Let me give you the explanation for this. So basically, you draw this matrix like this. Okay, when you are talking about one characters, but there are two contrasting or two phenotypes which has been involved. So this is a parent P1, and let's say this is a parent P2. The gametes which you define, okay, you define this as R. Let me make it simple. Let's say make it R1, and this is R2, this is R1 and R2. This will be an R1. This is one of, let's say this is a female and this is the male plant. So one of the egg will receive this R1 gamete, the other egg cells will receive R2 gamete. So we will write this, and let's say, let me make a correction. Let's say this is plant P1 and this is plant 2 P2 in this direction. Similarly, this plant 2 gametes R1 will write, and this is, we will write R2. Now, how to make this things clear, that how you make this process. So you take the first row and the first column, and then you just simply write capital R small r. Okay, and you keep your first row over here same, that is small r. Okay, or you can write either way, because it is ultimately coming this first row, and the second column it will be also capital R small r. Or then you will take the second row, first column, then it will be capital R small r, and you will also take capital R and small r. That's how you basically get all that is, you are getting R and small r as an heterozygous population over here. But since it is been found that all the plants appears round instead of a wrinkle, and this phenomenon is basically called or as a principle of dominance, because one is been dominant. So what does that mean also here? This also mean here that one of this chromosome gene, like say if I have to define this again with the same definition, that is capital R and this is small r, this one is basically whenever it is been present in single way, or whether it is present in a double way, so it is both the alleles is the same, then it will always be round. So this will be a round color of a plant or the seed shape. This will be also a round. But when in a chromosome, and let me again draw that part for you guys. So let's say this is a small r and a small r for this particular parent over here, they will appear wrinkle in shape of the seeds which will be. I hope this point is clear. Now let me erase all things to make you understand that what is a back cross and what is a test cross. Okay, so again taking example P1 plant, P2 plant, let's say this is a round, this is a wrinkle. In F1 generation, all the plant part will be capital R and small r. Whenever this F1 plant is crossed with either of the parent, that is P1 and P2, this type of cross is called as back cross. And whenever you make this cross, that is the F1 generation, with only the recessive parent, and in our case the recessive parent is P2, then it is called as test cross. So if I have to write this, that is the F1, if it is crossed with, let's say, P1, or a F1 which is crossed with P2, that is, if I have to define capital R small r genotype, which I am referring to this one, is been crossed. So this is the genotype of a P1 plant, this one. And similarly, this is F1, we know the genotype, and it is crossed with the recessive parent. This is called as a backcross. And when you take a F1 plant and you cross with the plant which is recessive, and how we know that this plant is recessive? Because in F1 generation, all of them appears to be round shape of the plant, or maybe tall plants, or maybe it is a yellow seed coat colors. So other plant which is not able to express, that is called as a recessive plant, and we basically made a cross, that is the P2 plant as this. This type of cross is called as a test cross. When we will be discussing about linkage in our subsequent lecture, I will try to explain this as well once more, that is what is back cross and test cross. In simple language you can understand, back cross is a larger set and test cross is a smaller subsets inside that. Okay. Now let's moving ahead. The principle of dominance, which you can see here, is being explained here as well. So this is Mendel's law of dominance. As you can see here, this is a change the color. So this is a female parent, this is a male parent. Here they took the seed color into consideration. So when they took this F1 plant, okay, say this is capital Y, small y, how they came to this particular conclusion, that is a plant P1 when it is been crossed with a plant P2 which is small yy. So in the F1 generation you will get capital Y and small y. Now when you do a second cross, means you will make this F1 cross with its F1, this type of cross is called as selfing or self cross, the same genotype individual which has been crossed. So they have shown here the gametes, as you can see here, capital Y, the small y, that is how the 50% the gametes which is coming, the capital Y and the small y, that is the first row, the first column, first row, second column, and then first row, second column. So now we are basically moving ahead to one of the other second generation, and that is the second filial generation. When we take a cross of this, now phenotypically, if you will observe here, these are three plants. Phenotypically they will appear as yellow, and one plant or the seed color appears to be green. Now coming back to try to explain this in the same example which we are actually doing here, that if we make a cross of a round seed shape of a plant, initially in F1 we find that all of them is a round, but in F2 generation we found that one plant is three plants in phenotypically they appear as round in color or round in shape, and one of that plant appears in a wrinkle. So the parental phenotype which was there, that is the P2, which was masked, or the recessive plant which is masked in case of the F1, they appears and comes in the F2 generation, and that is what is very surprising result which Mendel observed. And then based on this he basically proposed his principles of segregation. Okay, so in principle of segregation, if I have to define that, so it is been defined as or I probably will show you in terms of slide over here, that in the formation of the gametes, the paired hereditary determinants separates and segregate in such a way that each gamete is usually likely to contain either member of the pair. Means if you have a capital R and a small rr in the F1 individual, you basically get capital R and small r individual. But when you solve this, then in the F2 generation, you also receive, that is the capital R, one of this individual will be this, two of the individual will be this, and the other which individual will be this. And why it is appearing? Because there is no biasness. That's what Mendel is trying to explain in terms of principle of independent segregation. That is, whenever the gametes, when it is been forming, this gamete separation is independent phenomena. Okay, and that's why when you see in this particular case, over here has been shown that the genotypic ratio is coming 1 is to 2 is to 2, and the phenotypic ratio is coming 3 is to, means that is they appearing as the yellow seed color, or round seed shape, or a tall plant type, or say you can say the purple flower color, whatever you want to say. So this which is appeared in F1, that is dominant, but the recessive parents, they reappear in the F2 population, and this reappearance he basically proposed that it is due to the separation of the gametes, and this separation or the segregation of the gametes is independent phenomena in the process of gametogenesis. With this, I will stop here, um, and maybe in my next lecture I will continue this discussion of principles of segregation and principle of independent assortment and the dihybrid cross ratio. So if I have to summarize in today's class or in today's lecture what I have described to you about, so I have described to you about that there are certain definitions or the terminologies which we use in genetics, like alleles, the genotypes, what is phenotypes, monohybrid cross, um, what is backcross, which is test cross, and I try to explain to you this. I've also shown to you that what is the process of gametogenesis, a brief idea about a meiosis as well. Then we also discussed about what is meaning of a dominance, that is which has been appeared in the F1 generation, and which is not appeared, that type of trait we called as a recessive trait. Then we also discuss about that in F2 population, that this recessive trait actually reappear, and this reappearance of recessive trait is basically because the gametogenesis process is independent phenomenon there. Just an example over here, probably most of you know about the Harry Potter, okay, and the Hermione. This is a rolling of tongue in a human being. It is a dominant character. Someone who can roll the tongue, they are dominant, and someone who cannot roll the tongue, they are called as a recessive. Similarly, you can say the black eye color, in just for sake of a discussion in class, could be a dominant as compared to a blue eye color. So as I mentioned in my previous lecture, that is basically it's called as polygenic inheritance over there. And this is about the principle of independent segregation, and I will be explaining this principle of independent segregation in my next lecture. So thank you again, and I hope you get benefited from this, uh, my next lecture.